Thank you, George, for the invitation, first of all, to share my work with you and the community at Unique Medical School, and especially the students. Okay, so I'm thrilled to be here. I just started back in September to be part of the medical school and this growing research community. So I'm also delighted, of course, to be talking to you today about some of the research that I have done and that I'm planning to do here at the medical school. Since the audience is fairly diverse and I know there's a lot of students, I'm going to try and keep this as simple as possible so that you can learn something by the end of it, and hopefully learn something interesting by the end, and perhaps something that will spark your interest in developmental biology and embryology, which I'm interested in, and how we go about studying these fields and making new discoveries that will help physicians, hopefully maybe some of you in the future, treat or even prevent congenital malformations or diseases. And by congenital malformations, we mean anomalies that arise during development. So the baby is born with these diseases, and this is kind of like the focus on this talk and of my research. So today I will be talking to you about two different routes that researchers and personally I have used in biomedical research to understand the pathophysiology of certain diseases that arise during development. Both of which involve the use of animal models, okay? And I would like to start by emphasizing the importance of the use of different animal models in biomedical research. And since I'm a developmental biologist, I will focus on organisms that we use in developmental biology to understand human developmental processes that will help us understand, it will help us understand basic developmental processes that will help us understand human diseases, right? So this is the whole point of our research. So as you probably already know, we can't really use humans, right, as to understand this for ethical reasons, obviously. The most we can do with humans is human genetics. So we can find genes that are mutated in certain diseases, but then we need to use animal models to better understand the pathophysiology of those diseases, okay? So I'm gonna talk, again, as I mentioned, some of the models that me and others are using. So the mouse that you're probably all familiar with, it's been extensively used in developmental biology because, first of all, it's a mammal, just like humans. Its genome is very similar to the human genome. About 85% of the protein coding regions of the mouse and human genomes are identical. So we're pretty, very, very close. We can genetically manipulate them, and especially nowadays with the CRISPR-Cas9 technology, which you might have heard of, it's very easy to introduce the exact same mutations that are found in human genes that cause certain diseases. So we can make a mouse, for example, carrying the exact same mutation as a human patient, and I'll be giving an example of that in some of the work that I have done. So as you can imagine, this is very powerful because you can mimic the exact same mutation as in humans in your animal model. Then, let's see. So then we have the zebrafish, which is also a great genetic model. It produces a lot of embryos that are also transparent. They are outside, so they're external, so you can easily see them and manipulate them. You can see things happening live, especially in development. There's a lot of morphogenetic movements happening. So you go, from one cell to a whole organism. So a lot of things happen that you can easily visualize by just looking at the embryos or taking movies of these embryos and then going back. And it's also very useful because 70% of his genome is homologous to the human genome, meaning 70% of the genes are identical to human. Another model is the chick embryo, which it's a great model to use as for experimental manipulations of embryos. It's not that great for genetics, but has been classically used in development to watch things happening. Because as you can imagine, you have the egg, you have the egg, and you can make like a little window on the eggshell. You can remove the eggshell, see the little window, and you can watch the embryo as it's in the incubator growing. And this way you can also easily manipulate the embryo, the areas that you're interested in, and see what happens. Another one is the frog. So the frog has also been classically used in development. We've learned a lot about the different morphogenetic movements in development, about key movements in development that we still know now. Nothing has changed from a long time ago. And the frog, the advantages of this is that it makes a lot of eggs, a lot of embryos. They're external, so you can get the embryos themselves and look at them. They are very big, so you can easily manipulate them. It's not a great model for genetics, for genetic manipulations, but for experimental manipulations, it's a very good model. So these are the 4 vertebrate models that researchers have used mostly. In development, we have also used invertebrate models, such as the fruit fly. May come to surprise to you, but the fruit fly has been extensively being used to understand development. 60% is genome 60% identical to the human genome, and it has great genetic tools. So you can easily manipulate the genes and see and watch some processes, what goes wrong in some processes when you remove a gene or when you overexpress a gene, for example. And a few Nobel Prizes in medicine have been awarded to researchers actually that have been studying the fruit fly. For example, also one of the first genes related to autism has been identified using the fly. So a lot have been found using this little fruit fly. Another one is a worm. This worm called Caenorhabditis elegans has also been extensively used in research. This is a hermaphrodite, meaning that it produces both eggs and sperm, so fertilization can just happen in one worm. It's composed of 959 cells, and the fate of each cell, the developmental fate of each cell has been mapped. So back in the 60s, Sidney Brenner, one of the people that discovered this or brought this as an experimental model, sat in the microscope, and microscopy back then is not the microscopy that we have now, sat under the microscope and watched every single cell, every division, starting from one cell to all the way to the 959 cells, and watched every single cell, what happens to it. So it's a great model to study cell fate because it's already mapped. Until now, 3 Nobel Prizes have been awarded to researchers working on C. elegans, including the discovery of RNA interference, or RNAi, which is an endogenous mechanism used in animals to silence expression of genes, not on the DNA level, but on the RNA level. But nevertheless, very important mechanism that researchers have taken advantage of and used extensively in research. Also, for those of you that know GFP, green fluorescent protein, that was the first animal that was used in, and the researcher that did that got the Nobel Prize in medicine for it. So I myself, I'm very fond of this animal model because I did my PhD working on C. elegans. That's why I have a lot to say about it. But anyway, let's move on. And lastly, one of the models that is being used is this flatworm called planaria. Okay, so this is a relatively new model, but showed a lot of potential for understanding developmental processes and regeneration. So this little worm here, you can cut it into tiny, tiny pieces and it can regenerate the whole organism in a matter of days. Okay, so and it does that because as an adult, it still has a lot of pluripotent stem cells. So you can cut it up into tiny pieces and it will create the whole organism again. So this is a hot topic now, regeneration, because as you can imagine, if you can regenerate things, you can start regenerating organs, you can treat neurodegenerative diseases, and so on and so forth. So it holds a lot of promise. Okay, so the main goal of researchers in biomedical sciences, as I mentioned, is to find cures, treatments, and even better prevention of diseases, right? Of course, to reach that stage, you need first to understand what goes wrong on the genetic level. So if there's a genetic mutation causing the disease, then understand the pathophysiology on the cellular and molecular level, then hopefully find a treatment or cure, right? So you really need this what we call basic science first to understand these processes, and then you understand the disease on the cellular and molecular level, and you can go on from there to look for treatments and cures. So there's two main ways researchers go about doing that. So over here, so first, human geneticists will find the causative gene of a disease, and then researchers will take that and go to an animal model, introduce that genetic mutation or manipulate that gene and see what happens to the animal. If the animal can recapitulate the disease, for example, if you see defects in the animal that are the same or very similar as the defects observed in humans, then you have a very good model, and you can use that animal to try and figure out what goes wrong on the tissue, cellular, and molecular level. Now, the second way is a more experimental way where you're interested in a specific process, let's say craniofacial development. Then you go and manipulate genes that you think may be involved in that process, and if they cause a malformation, then you can, from there, you can learn more about the developmental process you are interested in, right? So for example, I'm interested in craniofacial development. I manipulate a gene in the mouse. I see a craniofacial defect in the mouse, and I say, okay, this gene has a role in the development of the craniofacial skeleton, and then you can dissect out its role using this model. And then, of course, human geneticists can take this gene, can take this information and screen for that specific gene in diseases where the genes have not been identified as the causative form. So, you know, it can go, you can use both ways to understand disease. So today I will tell you two short stories about using both of those ways in disease-driven research, right? So the first one, the study of vitamin B12 deficiency disorder, which we have very recently wrapped up and has finally been accepted for a publication. It involves the generation of new mouse models based on known human genetics, then the dissection of the pathophysiology of this disease using these mouse models, and then I'll tell you about it. There's a novel finding on a new aspect of the pathophysiology of this specific disease that has not been uncovered before, and we were able to uncover it. And then let's see. And then the second story involves a more experimental way I mentioned in trying to understand craniofacial development and craniofacial malformations using primarily the mouse. And this is a work that I have started before coming here, and I'm planning to expand here at the medical school. So let's start with the vitamin B12, also known as cobalamin deficiency disorder. Okay, so we all know how important vitamins are for proper development and homeostasis, right? We know that all vitamins, with the exception of a few, are essential. What that means is that our bodies cannot synthesize them, and so we need to obtain them from different food sources. Today, I will be talking about vitamin B12, also known as cobalamin, and I will be using B12 and cobalamin interchangeably throughout the talk, and it just means the same thing. Okay, so I will be talking about the role of this vitamin in development and disease. Okay, so we know that the intake of vitamins, we take our vitamins, but actually this is not enough. Our cells also need to have the proper biochemical pathways to convert the vitamin B12, whose original form is inactive, to an active form, right? So, and the active form of B12 is, in this case, is in two essential coenzymes that will go on to participate into important conversions. It doesn't really matter, the details don't really matter from this. But what's important is that we take vitamin B12 in our bodies, is in inactive form, then it has to go through these biochemical pathways that involves a lot of different enzymes and receptors, etc., to convert it into the active form that can actually do the job. Okay, and for example, B12, its job after it gets activated is to be a coenzyme that is involved in the production of an amino acid methionine, which is very important for DNA, RNA, and proteins and other stuff, and also in the production of energy. Okay, so okay. So this is the intake of the protein. What we also know is that components of these biochemical pathways are controlled by genetics, right? So all the protein in our bodies have instructions by genes to what they will become. Okay, so therefore, even if you have your cobalamin or other vitamin intake, you may not be able to convert that vitamin into its active form due to genetic mutations, any of these enzymes or other components of this biochemical pathway. So this is what I mean by saying nature and you need nature and nurture. You need nurture because you need the intake of your vitamins, but also you need nature, you need the right genetics, you need your enzymes to be working properly to be able to transform that inactive vitamin into its active form to do its job. Okay, so you need both. An example of a vitamin deficiency disorder that is due to a genetic cause is a disease with this very unfortunate name, methylmalonic aciduria and homocystinuria cblX type, thankfully also known as cobalamin or vitamin B12 deficiency disorder. Okay, so human geneticists have identified mutations in a gene called Mmachc. It's the cause of this cobalamin deficiency disorder. This is a rare disorder that affects about 1 in 200,000 born. It's a severe multi-system disease, meaning that it affects several different organs in the body, and is characterized by intrauterine growth delay, meaning the baby is smaller, doesn't grow as well, severe brain malformations, anemia, renal and cardiac defects, as well as renal degeneration. And importantly, like fortunately many rare disorders, the pathophysiology of this congenital disorder is largely unknown. And when I say pathophysiology, I mean the cause, what happens in this at the cellular level to cause this disease. Okay, so what is this gene Mmachc that is affected? You need to know about Mmachc is that it is an enzyme that is required in the cell to convert inactive B12 into its active form. Okay, so don't worry about this diagram here. Again, all you need to know is that, again, you get inactive B12, these red stars here. Mmachc, this enzyme will convert inactive to the active forms. Okay, so when you don't have properly functioning Mmachc, which is the case in this disease, you cannot convert your B12 into its active form. So even if you just keep taking B12, it will be useless in your body. Okay, so that was the first gene that was identified for that disease. Recently, there was an X-linked variant of the disease called cblX. And these patients, although they have a very similar cobalamin deficiency disorder, do not have mutations in MSHC, rather into other genes whose products control the expression of MSHC. And I will explain that in a bit. So these two new genes that were identified by human geneticists are this gene called HCFC1 and THAB11, also known as Ronin, and from now on I'll be calling it Ronin. So what is the relationship of these three genes? So we talked about MSHC as being the enzyme in the biochemical pathway. Ronin and HCF1 actually control the expression of this gene. So Ronin is a transcription factor, HCFC1 is a cofactor for Ronin. So the control expression of MSHC, and to remind you, MSHC is required for this conversion to take place of B12. The deficiency, in the cobalamin deficiency disorder, MSHC is not functioning properly, so you fail to make that conversion, right? So now in these new patients that have cobalamin deficiency but not mutations in MSHC, have mutations in either the transcription factor that drives the expression of this gene, or its cofactor is also required to drive the expression of this gene. So basically what happens in these other patients is that you fail to properly express MSHC, so you still don't have the proper, the functioning enzyme to make this conversion. Okay, so given that information, we decided to make mouse models of this new version, let's say, of cobalamin deficiency that is called cblX. So we used the CRISPR-Cas9 genome editing technology to make two mouse models. So we made an animal model, a mouse model carrying a mutation in HCFC1. I should say that this is the exact same mutation that the majority of the patients carry. So we were able to introduce the exact same mutation. And the details don't really matter of how we made it, but what's important is that we made the mouse, and it turns out that mutant mice are subviable postnatally. What that means is that some of them survive after birth, and some of them do not survive. They die before birth. Okay, so this is one, and then we made the second mouse model for the other gene that had a mutation causing cobalamin deficiency disorder in Ronin. Okay, so again, we use a CRISPR-Cas9 technology to make this point mutation. It's a single nucleotide substitution. It doesn't really, these details don't really matter. And these mice, homozygous mice, meaning that they carry both copies of their gene has the mutation, so they're homozygous. These die shortly after birth. Okay, and I'll be talking mostly about this mouse model to keep it a little bit simple. Okay, so now we made the mouse model. So now we need to see whether they recapitulate the patients, right? They recapitulate the defects that the patients have. And the first question we asked is whether the cobalamin pathway is affected, right? So we're modeling it after a cobalamin deficiency disorder. So we wanted to see, is cobalamin affected? Okay, and the answer is yes. I'm not going to the details of how we did this, but you can measure the inactive form of B12 and the active form of B12, and we're able to see that the active form of B12 was greatly reduced in these mutants. What this means is that yes, these mice have a deficiency in cobalamin. Okay, so after that, we started looking at different defects, the phenotypes in these mice to see again if they recapitulate the disease. And the first thing we notice is that this is just before these embryos are born. They are much smaller than their control litter mates. This is consistent with the intrauterine growth delay, so that the failure to thrive is observed in the patients that have cobalamin deficiency. Another thing that we noticed from these embryos is that they die as soon as they are born. And we figured out that they die because they cannot breathe. Okay, so they cannot take their first breath, so they end up dying after a few minutes, really like 10, 20 minutes after they are born. Right, so then we started looking at the different organs in these mutant mice that we know that in humans are affected, right? To see again if these mouse models is a good model to study this disease. So first we looked at the heart. Always on this side we have the control, and on this side we have the mutant. So again, all these are in the mouse embryo. So we're able to show that in the heart there is this defect in the capituli of the heart that resembles a cardiac condition called left ventricular non-compaction that affects the trabeculi and the cardiac, and therefore the cardiac muscle of the heart. Okay, so and a lot of cobalamin patients have been reported to have structural cardiac defects such as this. So again, this is it tells us that this recapitulates the human disease. We also know that a lot of the patients exhibit anemia, and especially megaloblastic anemia. And this is a type of anemia where you have less blood cells, and your red blood cells are much bigger than normal. Okay, and we're able to show that indeed these red blood cells are bigger in the mutant than in general red blood cells, and reduced in these patients, in these mice, again, recapitulating the defect that we observed in the human patients. Major aspect of cobalamin deficiency syndrome is the neurodevelopmental defects observed in the patients. And so we looked at the brains of these mice. We observed, again, this is the control, and this is a mutant. So for example, here, this is a micro-CT scan of the mouse embryo. And this is just looking at from the side, the sagittal view. So we observed hydrocephaly, or the accumulation of fluid in the ventricles. Here, as you can see, which is consistent, again, with some cobalamin deficiency patients. We also were able to observe thinning of the neocortex. So this part here is the neocortex of the brain, and it was much thinner than in the control. And so this suggests defects in neurogenesis. This is all I'm going to say about this now, and this is a phenotype that I am planning to look more into now that I'm here at the medical school, to see what exactly what's going on in the brain. If there are some defects in neurogenesis, we think that there may be something interesting going on here between the switch between neurogenesis and gliogenesis. But we'll look more into that hopefully soon. And lastly, we're able to see some craniofacial defects that were also observed in cblX patients. Example, so this is a control litter made here, so you can see the skull here, and this is the mouse. In the mutant, you can see that there's a deviation of the snout here. You can see it in the mouse as well here. So we call these mild craniofacial dysmorphias, but they are highly reminiscent of these two brothers here that had a cobalamin deficiency disorder. Again, showing that our mouse model recapitulates aspects of the disease. So to summarize this part, we made these two new mouse models, and we're able to show that they are deficient in cobalamin, just like the human patients, and they exhibit many defects in many different organs and tissues, very similar or very similar to what the human patients exhibit. So they're good models to study the pathophysiology of the disease. So now that we know that our models resemble this human condition, we wanted to learn more about the pathophysiology, what causes this disease on the cellular and molecular level. And very briefly, I will say that we performed some gene expression and proteomic analysis, among others, and we found that ribosome biogenesis is actually affected in these mice. And this is something, and this is a novel finding that we didn't know of before. Again, you don't need to worry about all these that I have here on the slide. Take-home message is that in the mutant mice, the ribosomes are not functioning properly. So we also did a lot of functional work to prove this that I'm not showing here for the sake of time. If anyone wants to talk about it after, of course, we can. And just to remind you, the ribosome is the machinery in the cell that translates all proteins, right? And is absolutely required for cell growth and proliferation. Okay, and again, if anyone wants to talk about it later, there's a big discussion going on about, if the ribosome is so essential in the cell, if you have a defect in the ribosome, how come you end up with these tissue-specific defects and not being able to survive if you had a defect in the ribosome, right? And there's a lot of different theories and ideas of why this is. And if anyone is interested, I can definitely talk about it afterwards. Okay, so we found that there's some defects in ribosome biogenesis. Now we know that there is a group of disorders called ribosomopathies in which ribosomes are affected, and patients can have tissue-specific abnormalities. Okay, so we looked in our mutant to see whether we find those abnormalities characteristic of ribosomopathies, and we indeed found several. So these very tissue-specific abnormalities include this white belly spot in the middle, right here in the belly. They include skeletal defects. As you can see here, these two are embryos, and this is an adult mouse. You can see that some skeletal defects here, this curling of the tail is due to some skeletal defects. These are some skeletal preparations. This is the cervical vertebrae here, and some characteristics that we also saw is this what we call homeotic transformations. So you get, for example, here we get a rib where it's a cervical bone. You're not supposed to have a rib here, and we get an extra rib. So these are called homeotic transformations, and they are very characteristic of these ribosomopathies. Also, exencephaly, we get some exencephaly, which is the exposure of the brain, okay, on the outside, and some polydactyly, so you get extra digits, okay? So, so our animal models really have shown us that there actually may be two aspects of the pathophysiology of this disease. So we knew of one, right? So we knew that there's less cobalamin. So the first one is the reduction of this key enzyme Mmachc that I talked about in the B12 pathway. The Mmachc that causes a reduction in the active form of B12 causes cobalamin deficiency disorder. Okay, so this is one aspect. And the other novel finding that came out of the study is that there is a reduction in ribosome function. So cobalamin deficiency disorder can also be categorized as a ribosomopathy, right? So as you can imagine, this changed the way that physicians treat patients. Okay, so currently the main treatment is cobalamin supplementation. So they give patients B12, vitamin B12, but it's actually not very effective in many patients. In some patients, it helps them, and in some patients, it doesn't really help them. Okay, so this, of course, could be because there is a ribosomopathy deficiency as well it has been unknown as of now. So again, this is the power of using animal models to decipher the pathophysiology of diseases like this. Okay, so I'm just keeping an eye of the time. Okay, so now I'm going into the last part of my talk which is about the craniofacial aspect of research that I'm very interested in and that I'm planning to continue here at the medical school. Okay, so craniofacial development is, of course, the development of the bone and cartilage of the head and face. And what you're looking here is a mouse embryo, and this is what will become the eventually head, the face and the skull of the mouse embryo. And this part, you know, in the first part, I told you about how we knew the human genetics. We knew the gene, the causative gene of that cobalamin deficiency disorder, and we used that to make the mouse models to decipher the pathophysiology of the disease. This part is the more experimental part I was talking about, where you're interested in a process and you're trying to understand it better so that you can go from there and understand diseases, craniofacial disorders, for example, a little bit better. Okay, so let's see if this will play. Okay, unfortunately, I don't know why, but this is not playing. Okay. Anyway, so it won't play, but this is actually a movie that I wanted to show of craniofacial development, just to show how that it's a dynamic process, right, which requires vigorous morphogenetic movements, that I was hoping of you to watch this and appreciate from this movie. And I'm just gonna briefly tell you what this movie was about, and I can share it later if you want. But this movie is actually it was made by taking 3D scans of a real human fetus during the first trimester. So this is what will become the head. And this is the midline here. You can see this is the one nostril of the what will be will become the nose. So this is the right side, this is the left side, and this is the embryonic, what we call facial prominences, that will give rise to the mouth, this here, the upper and lower lip, and the nose here. So what ends up happening is that you get both sides coming in the front and fusing right here in the midline, if you can watch my hands, fusing in the midline, forming our face, okay? So really our face is like two parts that come together, okay? And one example, you can see the lip here, this is where it comes. Anyway, so if anything goes wrong during these morphogenetic processes, one can end up with craniofacial defects. For example, cleft lip and palate happens because the fusion there doesn't happen properly. Okay, now, okay. So congenital defects, no, something happened to my mouse. Sorry after that, okay. So congenital craniofacial defects are a significant medical issue that affects 33% of all babies born with a birth defect, now, right? So these are some examples shown here. Some of the most common craniofacial defects are craniosynostosis and cleft lip and palate. Craniosynostosis is the premature fusion of your scalp bones. So usually your scalp bones don't fuse until about a year after you're born. And this is to allow flexibility of the skull for the expanding brain. So your brain still expands, gets bigger as you grow after you're born. So to allow for that expansion, and also to allow for the baby to go through the birth canal. Okay, so craniosynostosis is caused when these bones are fused prematurely while the baby is still in the womb. Okay, and it can cause problems in the skull. Cleft lip and palate, you probably all very familiar with, is the most common craniofacial malformation, and is caused by a failure for a fusion of the upper lip and sometimes the palate, the top of your roof inside. And then we'll have these more rare cases. Frontonasal dysplasia is caused by abnormal development of the facial bones. And Treacher-Collins syndrome, which I have actually extensively investigated when I was a postdoc, and this is due to hypoplasia of the craniofacial skeleton. So you don't make enough facial skeleton. Anyway, so now we know that there's a cell population called the neurocrest cells that give rise to the, I'm sorry, I'm having some trouble with my mouse. Anyway, so we know that neurocrest cells it's a cell population during embryogenesis that gives rise to the majority of the craniofacial skeleton by partially driving morphogenetic movements and also differentiating into the actual bone and cartilage of the face, right? So thus defects affecting the cell population during development underlie the majority, the cause of the majority of this craniofacial malformation. So I'm interested in studying the cell population to understand craniofacial development. Okay, so now what is the neurocrest? I will very briefly, running out of time, very briefly tell you a little bit about this. First of all, it's I think the coolest cell population in the body. So the neurocrest is a transient multipotent migratory cell population that arises from the dorsal part of the neural tube from a pool of neuroepithelial stem cells, and then it delaminates through epithelial to mesenchymal transition and migrates throughout the whole body of the embryo, right? And upon reaching its destination, then it will differentiate to the proper cell type. So this you can see a mouse here at around embryonic, this is around embryonic day 9, and this is a day later, just to give you an idea, the mouse embryos are born around embryonic day 18.5, so this is kind of like halfway before they are born. So in green, you can see all the neurocrest cells. So this is the head of the embryo here, it goes down the trunk. And this is here, you can see better that all these neurocrest cells came from this part, the dorsal part right here, and they migrated all around the embryo here. And you can see there's extensive migration in this craniofacial area right here. And this is because it will make most of the craniofacial bone and cartilage. Now, perhaps the most astonishing aspect of neurocrest is that it is multipotent. What this means is that it can give rise to a wide variety of cell types, as shown here, including neurons, melanocytes. For the purposes of today's talk, it will give rise to the majority of the craniofacial skeleton. Here is a schematic of a mouse skull and a human skull, and all the collarbones are derived from the neurocrest. Okay, so it has a major contribution. So we know a lot about neurocrest cells, about the different stages in the development, but we don't really know much about the reason they're multipotent. Okay, so what makes them multipotent? What is the transcriptional regulation of this multi-potential nature that they have? So I'm interested in figuring that out. Okay, and I have a candidate gene that I think may be involved in this. And this gene is Ronin. It's the same gene that I was talking to you about in the previous slide, in the previous part of the talk. And what I've done is to, what you do in these cases, you can conditionally, that means you can knock this gene out just in neurocrest cells, okay, to avoid any lethality. But if you take it out early, then the embryo dies, right? So you take it out in the neurocrest cells only, and you study and see what happens. So when we do that, we get an embryo that is missing all the facial skeleton. And all this skeleton is derived from the neurocrest, okay? So this is a very dramatic phenotype, which is a very cool phenotype to see. And long story short, this is kind of where we are right now, and we're trying to figure out what goes wrong, and we're trying to go back in the embryo, in the early embryo, to see when this defect starts. And the only thing I'm going to say about this slide is that we're using a lot of different techniques, including immunohistochemistry, immunofluorescence, in situ hybridization, skeletal preparations, to figure what goes wrong in these cells when you take this gene out, okay? And with that, I'm gonna end here. This work, we've had a lot of collaborators for this work. They're all listed here. This work was mostly done when I was a research associate in the Poche Lab at Baylor College of Medicine in Houston. Again, these are all of our collaborators. And with that, I will leave you with these cool movies here, and I'll take any questions that you may have.
Thank you, Annita, for a very interesting presentation. We have few questions. What I can do is to open the mic to those participants who would like to pose their questions instead of us reading those questions. But may I start with one question, please? You mentioned at the very beginning the percentage of homology of different animals to the human genome, but I'm not sure if I missed the one about for frog. What was the percentage of the frog?
You noticed that. So for the frog, so there's what I could find is that there's about 75% homology to the genes that are known to be involved in human diseases. So all the genes that are known to cause human diseases, about 75% of those are identical in the frog.
All right.
Makes sense. Otherwise, I couldn't find the information of, in general, what's the homology. But this is pretty significant degree of homology also.